for coming, everyone. Just a brief update on the title, as you may have read. So I changed the title to Evidence for In Situ Iron Cycling from focusing just on the lithoautotrophs, because I think the iron cycle in general makes for a more interesting story in my system. So to get everyone oriented, Chocolate Pots is in Yellowstone, and it's in the northwest corner right along the Gibbon River, denoted by the blue X. And here we have a couple pictures of the hot spring. We have the, oops, there we go. So we have the vent pool up here, the flow path. And then Chocolate Pots is a very interesting hot spring because it's circumneutral pH, mildly thermophilic, and contains a high amount of iron and silica in the spring water. From an astrobiological perspective, we're interested in environments like this because of the new and recent discoveries of circumneutral pH-like environments on Mars and the evidence of relic hot spring-like features. And if we're able to nail down evidence for internally cycling iron-based metabolisms in environments like Chocolate Pots, then this could, in theory, be applied to some of these relic features on Mars. So we have two main hypotheses going forward with this study. The first is that there are native, active iron-reducing microorganisms present in the Chocolate Pots vent, and also that there are lithoautotrophic iron oxidizers, which are also actively cycling iron. And in addition, as lithoautotrophs, they are fixing fixing CO2 and able to supplement other sources of carbon for the heterotrophic community. So backing up a little bit, some of our initial work with materials from Chocolate Pots, we ran a series of iron-reducing enrichment culture studies. And this is a graph showing the repeated transfer of these cultures for over 4 years. And they've been able to sustain a fairly high level of iron-reducing activity. And through both 16S and metagenomic sequence data, we've identified sequences that are related to known iron-reducing taxa, including Geobacter and Melioribacter. Moving one step towards teasing apart the in situ environment, we ran another series of short duration incubations using just the native sediments and amended with and without additional electron donor. And the things I'd like to point out here is in this feature, which that figure, which corresponds to material collected from the hot spring vent, you have an almost equivalent level of iron reduction activity regardless of electron donor source. Whereas if you move a few meters away at site 3, there's still a measurable amount of activity of the organisms using their native carbon source, but it's greatly reduced. So this led us to the question of what is this internal carbon source? Is it coming from chemolithotrophs? Is it from the cyanobacterial community? Is it breakdown of plant or animal detritus from the external environment? Where's the carbon coming from? So, and we have 16S data from this as well, and several OTUs related to an organism, Acknowlements. While it is not implicated as an iron reducer, it is very similar to this Melioribacter, which I identified in the enrichment cultures. Taking a look now at the in situ environment, we collected a series of core samples moving down the flow path. And the 16S data from these samples revealed sequences related to known lithoautotrophic iron oxidizers, sideroxidins, and again, this relative of a potential iron reducer, Acknowlements. So the metagenomic workflow I'll cover in brief. You start out with a very complex community like Chocolate Pots, get all the DNA, sequence it, put it back together, separate it out into bins, which are you can think of as being different taxa within the community. And then what we're really interested in is what is the metabolic potential of some of these bins or taxa. And we're taking a targeted metagenomic approach looking at which genes appear to be involved in iron cycling and which ones are involved in carbon fixation. So here we have the community of 2 metagenomes that I assembled from chocolate pots. One is from water filtered from the vent pool, and another one is a combined assembly from 3 of those sediment cores that I mentioned. And once again, you see bins related to this known lithoautotroph and this possible iron reducer. So thank you to Dave Emerson for the lovely introduction to situ. I will not be covering that in my talk because I found no homologs of that in my system. But I will be talking about this other electron or extracellular electron transfer system, a porin-wrapped version where you have a porin, an opening in the outer membrane of the cell, and a cytochrome which fits into that and is able to transfer electrons into or out of the cell depending on whether it's an iron oxidizer or iron reducer. And there's 2 homologs in particular I want to point out. There's this MTOA system which is identified in sideroxidins, and there's this geobacter-like porin cytochrome system, and it's also been identified in that Acknowledgebacterium organism. These are just several different search tools that you can use, hidden Markov models, BLAST-P homology, many different ways to identify these genes. But one of the more important things that we're interested in is the genomic context of these genes, that genomically you need to find a porin encoded next to one or several of these cytochromes. Okay. We're looking for multiple CO2 fixation pathways, but for the sake of brevity, I'm only going to cover Rubisco today. So to quote my advisor, if you identify a bin with an EET system and Rubisco, that's pretty good smoking gun of an organism that may be involved in lithoautotrophic lifestyle. And we use very similar search parameters for these genes. So here I have a summary of all of the metagenomic bins from these 2 assemblies. In red, we have all of the bins that contained some model EET system. In yellow, we have all of the bins that contained a hit for Rubisco. And what we're mostly interested in is these bins that contained both systems and may be involved in chemolithoautotrophy. And then down here at the bottom are bins that were not involved in any of it. So bringing this back to the 2 organisms that I've been mentioning throughout this talk, the sideroxidins and the Acknowledgebacteriales, I just wanted to summarize the results in terms of those 2 organisms. So up in gray, we have the published genomes of these organisms. And again, sideroxidins containing this MTO system and Rubisco, and the 2 related Acknowledgebacterium and Melioribacter containing only that geobacter-like PCC system. Whereas looking at the 2 metagenomes, here we have a sideroxidins relative that all of a sudden has a PCC system. And we have these Acknowledgebacteriales that have MTO and Rubisco, which is really interesting that sideroxidins, which is a known lithoautotrophic iron oxidizer, contains PCC, which so far has only been implicated in iron reduction. So perhaps this is a relative that is able to carry out both iron oxidation and iron reduction. And for the Acknowledgebacteriales, as I mentioned before, has an EET system and Rubisco, so perhaps it is capable of carrying out a lithoautotrophic metabolism. So to wrap things up, once again, we have a highly active dissimilatory iron-reducing community at the Chocolate Pots vent, and that is able to sustain itself using only the native carbon source. We have metagenomic bins that are related to known and potential iron cycling microorganisms. We have the positive identification of homologs of genes involved in both these EET pathways and carbon fixation pathways. And as a side note, we're in the process of identifying lipid biomarkers from these materials to try and nail down a possible source of the carbon that's present in situ. And one last thing I would like to mention, so I only covered very briefly the model systems that we've been looking at, but we've also expanded our search to look for non-model EET systems, and we're also looking at all of the known carbon fixation pathways in these systems. And with that, I'd like to thank the organizers for the conference, my funding, my committee members who have helped with this project, Eric and Eric, and my colleagues back at UW-Madison, particularly Xiaomei He, who without her metagenomic expertise, we would not have been able to do any of this. So with that, I think I have time for a couple questions.
So we definitely have time for a couple of questions. Tim. Yeah.
Yeah, I really enjoy the Yellowstone talks. They're really interesting. It's a cool area. So what non-model systems do you have in mind exactly?
As far as the EET goes?
Yeah, exactly, yeah.
So again, a lot of this credit goes to Xiaomei and her computer programming skills. But we have scripts that we're able to search our metagenomic sequences for the heme binding site motifs, and any genes that have that appear to be multi-heme cytochromes, we then look for genes proximal to that that are predicted to be an outer membrane porin. So ones that aren't homologous to any of the models, but still seem to fit that same type of system with a porin and a cytochrome.
What about the E-pili or geopili? Any considerations about that?
Yeah, so that's something that I've considered and we've discussed as a lab quite a bit, but something that I still need to investigate with these systems.
Yeah, I agree with that.
Yeah.
Cool. Right, thanks.
Yeah, welcome, thank you.
So we still have time for another question if there's something burning out there. No? Okay, so let's again thank Nathan.
Okay, thank you.